that she does. And when you actually look at her bio, you see that she tells you more about herself than most researchers do, which I really like that. So anyway, Meghna, are you ready to go?
Thank you to Bill and to everyone at JCU for giving me this opportunity to present my work. I'm quite excited to be able to share my research with you all, what I've done so far. And unfortunately, it is over Zoom, but the good thing is I don't have to sit on a flight and go all the way to Cairns for this. But hopefully at some point we'll meet in person and carry on some of the discussions that I hope we can have today. Briefly, I'm a scientist at a place called the Laboratory for Conservation of Endangered Species and I live in Hyderabad, India. And I try to understand how ecological communities are changing in the times that we live in today. Can you see my slides move? Just checking.
Yes.
Yes.
Okay, great, thank you. So let's start with the obvious, right? Globally, ecosystems are experiencing dramatic changes. And unlike perhaps any time in our geological past on this planet, it's not massive volcanoes or giant asteroids that are mediating these changes. It's one species, us. Humans, in their quest for space and resources, have irrevocably changed planetary systems, and which has perhaps imperiled the future of biodiversity on this planet. So global environmental change is actually disassembling ecological communities as we know them, or as we have seen them so far. Now, to understand this process of disassembly, we actually need to start from the basics of understanding how ecological communities come to be in the first place, or how do they assemble. And there is a large body of ecological work that looks at this problem, in which ecologists seek to understand, okay, when we see a community at a place, why is it that some species are able to get to a place and why are some others not? What environmental conditions allow some species to do better than others, give them a competitive advantage, and maybe even exclude other species from being in a place? And within a community, what are the species interactions that drive the trajectories that we see in their populations, the way they perform in these communities? In other words, why are species found where they are found? What governs these factors? Why are some species common and other species rare? And what are these mechanisms that perhaps allow some of these rare species to persist, that generate and maintain an ecological community and allow biodiversity to persist? Now, this has been the focus of ecological science, biodiversity science, for a long time, but perhaps this understanding of community assembly has become even more important in today's day and age, where we are losing communities and losing species from the systems that we know at an alarming pace. And among the many drivers of biodiversity loss that I outlined in the previous slide, habitat fragmentation, which is the breaking up of once large contiguous blocks of a habitat into smaller parcels, is among one of the top global threats to biodiversity. And here in this picture, I illustrate this with forests being broken up by mines or roads or agricultural uses. But fragmentation is a problem for many different ecosystems. And the reason I put up this slide is because I study the impacts of fragmentation in forest ecosystems, and which is what a lot of my talk today will be about. But many of the concepts and theories are perhaps applicable to other systems as well, even though there will be specific problems and specific ways in which you might want to look at these other systems. But coming back to forests, there's been a long history of research in fragmented forests, and having Bill in the room is a bit disconcerting for me with all the stuff that he's done in this regard. But long-term studies from fragmented forests that have been monitored in, for example, in Brazil, at various parts of Brazil, we know that a loss of tree diversity in particular happens near forest edges. And these patterns, not just from long-term studies, but across the world, these patterns have been well documented. But it so happens that the mechanisms underlying these patterns are still being understood. And we don't still have a good handle on, okay, why is it that some species appear to do well? And what exactly are the processes that allow the predominance of some species and promote the extirpation of others? And in particular, we don't know the mechanisms that might be driving this loss of biodiversity that we see. So this is what sort of got me thinking, okay, we have this entire body of knowledge where people are talking about, plant communities are a great model system for understanding community assembly, why diversity is maintained, and we have all of these theories. Can we translate them to understand what happens, or how do we explain the loss of biodiversity near the edges of fragmented forests? And one such mechanism that promotes diversity is through biotic interactions between plants and their enemies. And it turns out that these diversifying interactions are the strongest at the earliest life stages of plants, which is seeds and seedlings. And there's ample data now to show that what happens during the seed and seedling stage can have implications for the diversity of the forest many years down the line, particularly when we are talking about these wet or humid tropical forests, which doesn't have much of a seed bank and really relies on the new seedling recruits, as we call them, the young seedlings that come up to maintain diversity through the long term. And this process of how this happens was encapsulated long ago in what we call the Jansen-Connell hypothesis, and I'm sure many people in the room have heard of it. But the basic premise of the Jansen-Connell hypothesis is that species limit themselves more than they limit other species, or more than other species limit them, right? Which means within species competition is stronger than across species competition. And this manifests as something called conspecific negative density dependence. And just to break down this term, this means that your population growth rate will decrease, or your per capita performance in terms of survival or growth will decrease if you're next to another individual of the same species as opposed to an individual of another species, right? And so the more abundant a species gets, the less well it will do in a population, which in a community, which allows the rare species to persist and hence maintains diversity. Now, Jansen and Connell proposed that it's actually the enemies of plants, pests, parasites, pathogens, that allow this kind, or that promote this kind of intraspecific, conspecific negative density dependence, and particularly host-specific enemies. So when you're if you're a seedling that's next to a parent plant of your own species, then you're more likely to be in an environment where your pests and pathogens that attack you have built up. So you will get attacked and you might die, but another species will do well. So this kind of enemy-mediated density-dependent effect actually allows species to coexist, opens up recruitment space for rarer species in particular, and maintains diversity in the long run. So we might extend that to then hypothesize or then surmise that if there are conditions where either enemies decrease or if their efficacy decreases, then we might have a situation where some species the dominant species won't have a check on, being dominant, and rarer species may get knocked out of the system to then decrease diversity. And this is something that I examined in the context of edge effects in forest fragments. So there is data, or there is information that suggests from just the world of fungi and insects that it is seen that specialized enemies may be replaced by more host generalist feeders in forest fragments, in human modified habitats, particularly near forest edges. We don't quite know why this happens, but there is some evidence that this happens. Now, this means that the specialization that is needed for the Jansen-Connell effect to operate may be lost in these fragments near the fragment edges. And also, when we look at an interaction with the abiotic conditions, warmer, drier conditions near edges can decrease the efficacy of some plant enemies, such as oomycetes, which again, recent studies seem to suggest might be a big driver of these density-dependent Jansen-Connell type effects. But they may also increase the activity of some enemies such as insects. And so they may flip the balance of which enemies are doing what, and therefore change the diversity and composition of the seedlings that end up recruiting at a place once these enemies act on them. So these are some of the kinds of questions that I thought I would examine in a human-modified landscape in the Western Ghats biodiversity hotspot. Now, for those who might not be familiar with this, the Western Ghats are a 1,600-kilometer long mountain chain along the west coast of peninsular India. I don't know if you can see my cursor moving, but it runs like this. My study site was located somewhere in the middle of this. It's a typical human-modified landscape in these systems, which is about 3,500 hectare of matrix of forest, tea, and human habitations. About 60 to 70%, 65% of it is still forest. And as you can, what I did was I went into these forest fragments that you can see here, and along a gradient of the edge, forest edge to the interior, I set up a series of plots, a bunch of plots that come together to form what we call a station, a series of these sampling stations to understand what kind of seeds fall into this area and what seedlings recruit from this area. So here you see that we put up these seed traps to capture the seeds that come into the area and then monitor what kind of seedlings recruit from these seeds. But in addition, we had 3 other treatments. We had 3 plots adjacent to the seedling plot, one of which we sprayed with a fungicide, another with an insecticide, and a third one where both these natural enemies were suppressed using biocides. The idea was to see, for the same community of seeds that arrive at a place, do we see a very different seedling community without the effect of these enemies? And do these effects vary from the edge to the interior of the forest? Now we pulled this data together we measured this for about a year, a little over a year, which was about 2 recruitment cycles for the seedlings, which means there were 2 rounds of seed fall occurred throughout the year, although there was a peak, but there were 2 rounds when seedlings recruited. And the first thing we checked was, okay, what are the patterns of diversity that we see in both the seeds and seedlings? Because seedling diversity might just reflect the fact that there's more seed diversity in some area, right? Now, what we found was, when you look at panel A to your left, I'm hoping this is to your left also, it's to my left. But seed diversity doesn't seem to vary substantially as we move from the edge, which is here, to the interior of the forest. But when we look at seedlings, we see that the low consistency, low diversity that we see until about 60 meters into the forest edge changes quite dramatically and significantly when we go about 100 meters into the forest, which has much higher diversity of seedling recruits than until about 60 meters. Now, the question is this related to the activity of these enemies such as insects and fungi that we spoke about? And for this, we looked at what our insecticide and fungicide treatments did at each of these distances from the forest edge, when plots were at each of these distances. And again, when we look at the interior-most conditions, if you would just look at this panel labeled E3, we find that the high diversity, or the diversity of seedlings that we saw, dramatically and substantially decreased when we sprayed a plot when we suppressed insects and fungi in a plot. And when we look at these other 3 categories, distances from the edge, we had low diversity, and this diversity didn't seem to be affected at all by the action of enemies, suggesting to us that insects and fungi did not seem to be able to regulate recruitment in such a way, or play a role in diversifying or modulating the diversity that we see until about 60 meters from the forest edge. And the next question is, so was this because they couldn't modulate recruitment or the way the seeds transition to seedlings? And the seed to seedling transition can have 2 components. One is just what is the baseline rate of a seed becoming a seedling, but also for each species, by how much does that rate decrease? The probability of transitioning to a seedling decrease as the seed number increases, which is the density-dependent Jansen-Connell effect that I referred to earlier, right? So in this graph on the y-axis is density dependence, which the more negative the number, the stronger is the density dependence, or the poorer you will recruit once your seed densities increase, right? Now, if you look at from the edge to the interior and you just look at the black dots and bars here, we see that the strength of density dependence in control plots where nothing was done does tend to be stronger in the forest interiors, the interior-most forests. And in the interiors, this strength of density dependence changes the regulation, when we suppress insects and fungi, indicating again that this diversity effect that we saw in the previous slide seems to be correlated well with the effects that we see in how insects and fungi are mediating the seed to seedling transition dynamics. And when they're not able, in situations where they're not able to control the transition dynamics, even though there is density dependence that occurs, when it is not occurring due to these more host specific enemies, we are seeing that it doesn't seem to have a diversifying effect. So maybe we still have rodents and some other mammals that are eating these seeds and causing some density dependence that we see here, but this is not resulting in a diversifying effect unless this seems to be mediated by insects and fungi, which are known to be more host specific. I just want to ask if anyone has any questions, please feel free to stop me and ask, because as I said, I can't see anyone. So just let me know. If someone could let me know if there's a question, that would be great. Okay, I'll continue then. So this effect that we saw on species diversity, we were also curious, is this in any way going to be related to the kinds of species that seem to come in, which is basically the traits of species. Now, one key trait that people have looked at in fragmented forests is the size of seeds, because we've seen that closer to the forest edge, we have smaller seeded species doing much better, or we have lower diversity of, the seed sizes that come in. The question is this in any way related to the activity of insects and fungi? And to some extent, the results are a bit, variable, but to some extent, we see that yes, just like the patterns for species diversity, these black dots show that functional diversity, or the variety in seed sizes that we see, does indeed increase when we go to the interior-most parts of the forest. But this is, I'm sorry, why, I don't know why the edge bar is not coming up. Sorry about that. But panel H is the one that indicates the interior forest conditions. And in these conditions, when we suppress enemies, this functional diversity comes down, an effect which doesn't happen as strongly and definite at, up to about 60 meters from the edge, and definitely not till about 20 meters from the forest edge. And we find that these changes in functional diversity are actually related to the extent to which smaller seeded species appear to be regulated by insects and fungi. And there is theory behind this. The smaller seeded you are, the more likely you can be more vulnerable, by and large, to insects and fungi, because you're investing in a life history that's not about defense, that's not about staying in there, but it's about just dispersing well and reaching lots of places and quickly establishing yourself. So that was the kind of idea that we went with. And in this graph, the black lines are indicating the recruitment, or the number of seedlings that recruit into a 1 meter by 1 meter plot, as a function of seed size, which is on the x-axis. And if you look at panel H, we see that there is an effect of, the black line tells us that larger seeded species tend to have better recruitment in these interior conditions. But up to about 60 meters from the forest edge, in these three panels, we find that smaller seeded species are clearly having much higher recruitment rates. Now, what happens when we suppress the enemies that might be mediating this recruitment success? We find that in the interior forest, which is the interesting result that we are seeing here, that allowing, or suppressing enemies actually allows a much higher recruitment rate for these smaller seeded species when compared to the larger seeded species that don't seem to gain as much, right? And this difference just doesn't seem to come up when you are up to about 60 meters from the forest edge, suggesting again that there are just like with or without enemies doesn't make a difference. The enemies are not making a difference to what recruits or what determines recruitment numbers up to about 60 meters from the forest edge. And this might be one of the reasons why we are seeing small seeded species escape and do really well in these conditions of, closer to the forest edge, explaining why their numbers seem to increase so much. In addition to the fact that there might be some light-loving species that increase, smaller seeded species seem to increase because there are no enemies or because there's poorer effect of enemies. And this might explain why this effect stays in these forests, right? These fragments are all about 100, 120 years old, and this is what is happening here today, right? But that brings us to this question that this is one study for 1 year, and over one small life stage. Does it even matter? And I was curious about this. And so I to understand this question of what might be the long-term implications of recruitment for diversity, plant diversity in fragments, I approached this problem through a framework called the modern coexistence theory. Now, in this framework, which is basically based on Lotka-Volterra models and pairwise interactions, but very simply put, two species would coexist if the differences in their fitness, which is just baseline, how well do you do in a given condition, are less strong or weaker than some of these stabilizing mechanisms such as density dependent. Now, by stabilization, I mean that any mechanism like the Jansen-Connell effect, which takes away from a species' advantage of being abundant, or reduces their population growth rate when they are abundant, or the flip side is it increases the population growth rate when you're rare. So stabilizing mechanisms have to be stronger than fitness differences to allow species to coexist. So we see that whenever this takes place, when there are strong stabilizing mechanisms, you will have higher diversity, or these systems can maintain higher diversity. So we took the data on seed to seedling recruitment from what we had, and I teamed up with a theoretical ecologist to model the future of, the future plant diversity that we might see, the plant community that we might see, if we pull out these parameters from the models for species based on their ability to recruit from seed to seedling. Now, there were two components, like I said. One is the baseline survival, which we assumed was a reflective of baseline fitness in different habitats, that is going from the edge to the interior of the forest. And the second component was the strength of negative density dependence, or think of it as the stabilizing effect. To what extent does a species limit itself? And we saw how that varied for about 18 species from the edge to the interior. We took all of these values together and then pumped them into the simulation model, which my collaborator Simon Stump was so good at doing. Simon is a great theoretical ecologist, and we really enjoyed working on this project together. And in this simulation, we pumped all these numbers in, and then we modeled, what does this forest look like many, many generations into the future when these are the recruitment dynamics that are likely to play out. These are the numbers that are governing recruitment dynamics, right? And there was a lot of math that went in, a lot of modeling that went in, but the basic premise, what we understood from here, is encapsulated in a metric called the invader growth rate. Now, in coexistence theory, invader growth rate means what is your ability to maintain a positive population, greater than 1 population growth rate when you are becoming rare. If that number is greater than 1, then you're going to stay in the community when you're rare. When that number dips below 1, it means you're going to go extinct, right? And there's some variability around this. So in this graph, each line represents a species, and each dot on the line represents its invader growth rate in different sized fragments. And there are 3 contexts to this, right? One is you have a fragment with an edge and an interior, and how species are behaving in the whole fragment. You have an intact forest, which is, we just take the values from the interior of the forest, and then we use that and say that this is what happens for the entire piece of the fragment. And then we have a fragment in which there is no such interior, which means imagine if edge effects were to prevail everywhere. Now, this is a thought experiment, but what this is going to tell us is if we have fragments of a certain size, but we don't have the habitat that can allow some of these stabilizing effects to take place, do we have species with net, can they maintain net positive invader growth rates? And you'll see from this graph that whenever you have these empty circles, it means a species is going extinct. So you see from these lines that when we have these fragments without interiors, most species seem to be going extinct. The proportion of species that are extirpated are much larger if you don't have these interior quality habitats that are able to support species coexistence, right? And we found that this was primarily related to the stabilizing effect of density-dependent regulation by enemies. So what this modeling tells us is that depending on fragment size, what is the effect of density-dependent mortality to shaping this invader growth rate? Now, this is based on a mathematical decomposition, which we, of course, don't need to get into, and I'm probably not the best person to explain it anyway. But very simply put, what we see from the blue line, from these three lines, is that when we compare these two graphs, baseline survival didn't seem to have much of an effect on explaining the variation in invader growth rates between these three contexts that we set up of intact forest, fragmented forest, and fragments without interiors, and did not seem to make a difference to what happens to invader growth rates or the ability of species to stay in a system when you have fragments of different sizes. But in all cases, it was actually the stabilizing effect of density-dependent regulation that seemed to explain whether a species is going to stay. What is its likelihood of staying in the community when in these different contexts? So the main take home for me from this study was to actually speak to a debate that has been happening about habitat quality versus quantity, where there is a habitat quantity proposition which states that it doesn't matter if you fragment habitats, but as long as there's adequate amount of habitat that remains, you'll still maintain diversity. Or, well, they say species richness, but here we see that when we are losing species in these different kinds of scenarios, what we find is that no, quality matters. And we have defined quality as the ability of habitat fragments to retain certain key mechanisms that allow species to coexist. And so if you're going to have what some of our data shows and our simulations show is that if you have a large enough fragment, but if the quality is not adequate, this is not going to support ecosystem, the community diversity. Sorry. So I hope, just wondering if there are any questions about this so far, because I'm going to shift gears a little bit here.
Can you,
okay.
Can you hear me? Yes. Sorry, yes, we'll do. My name's Nina. I'm helping organise the seminars, and we'll do questions at the end. I'll sort of take the mic round so that everyone can hear.
Okay, perfect. Yeah, that's good to know.
Thank you.
So, all right. Yeah, thanks, Nina. Okay, so we spoke a lot about fragmentation, and studying the future of biodiversity is, in forest fragments is something that drives a lot of my research. But like I mentioned, we live in changing times, and there is massive changes that are expected to the climate regimes that we see in different places around the world. And for plants and tree communities in particular, the extent of climatic water availability, or what we call seasonal drought, the amount of the dry period that a plant community experiences during the year, is known to be a huge driver of plant species diversity, composition, and community dynamics. And with projected changes in temperature and precipitation gradients, in many places, seasonal drought regimes are expected to change and intensify. And along that is predicted that plant diversity will sort of follow along and respond to some of these changes. Now, in order to understand how plant diversity at a place is, or the plant community at a place is going to respond to a change in seasonal drought, we first need to know, okay, how is extant conditions of drought shaping the plant community as we know them? So this is something that I wanted to do at a large spatial scale across the entire Western Ghats. And as I mentioned to you, this is a mountain chain that runs along the west coast of peninsular India. And what is interesting is that the monsoon winds actually hit the southwest monsoon, it's called, and it hits from the southwest, obviously, and drops a lot of rain in the southern part of the Ghats and then progressively decreases as it goes up. But interestingly, because of changes in orography or topography, there are some parts in the central and northern Western Ghats also which experience a high degree of rain. But what changes consistently is the number of rainy months. The south experiences about 9 to 10 rainy months where you have more than 100 mm of precipitation, whereas all of the rain that is received is concentrated into 3 to 4 months or even shorter as you move further north. So we see that there is this strong gradient in seasonality, and previous studies have shown that for both trees shown in triangles and shrubs shown in these circles, seasonality seems to be a strong driver of plant diversity. Now, why should diversity decrease with seasonality? There are two hypotheses. One is the physiological tolerance hypothesis, which says that drier climates limit some species because they can't tolerate those areas, but they will still remain in the wetter or less seasonal climates. On the other hand, you might have biotic interactions, which say that, okay, there are these species that do well in dry climates, but they're limited from being in these less seasonal climates because there are biotic interactions such as between enemies, or, there are environmental conditions that are not suitable for these dry affiliated species. So I sought to examine these two hypotheses, particularly the physiological tolerance or the implications of the physiological tolerance hypotheses at a large spatial scale. So I pulled all of this data together from a bunch of plots, more than 20,000 trees across the Western Ghats, and modeled the distributions of approximately 183 species for which we had data to see how they respond to a gradient of climatic water deficit. The more negative the number on the x-axis, it means the greater the climatic water deficit or more intense the seasonal drought. And what you see here is, as expected, species have a variety of responses, but there are two components to this model that are important. One is, and what you need to keep in mind mostly, is what is called a first order response, which is that is a species likely to be affiliated to a drier condition, which means that its probability of being in a place is going down as we move towards the wetter forest, or is a species more likely to be affiliated to a wetter condition and its probability of occurrence goes down as we move to a dry condition. So a positive value of what I call this first order coefficient means that it is wet affiliated. A negative value would mean that it is dry affiliated. And we found that species showed a variety of responses, and there were but by and large, most species in this area seem to be wet affiliated, and which is not surprising given this is a primarily wet tropical forest. But there were many species which clearly had drier affiliations. Now the question is, could they be, or could they remain, or could they form the communities in these wetter parts? And to do that, I sort of moved from the species, or integrated what we see at the species level into what is the pattern that we find at the community level. And there we see that if we look at the mean of this first order response, which is this affiliation towards a dry or a wet condition, we find that as we move towards a wetter forest, which is here, if you can see my cursor moving, a low value of climatic water deficit, we find that the mean first order response, or the mean affiliation, is more towards wetter conditions, and that dramatically decreases as we go into drier forests, where it is comprised primarily of species that seem to be affiliated towards dry conditions, suggesting that these wet affiliated species seem to be getting knocked out of these drier systems. And when you look at the variability, that shows us something interesting as well, which is this whole thing about getting knocked out, right? If you are in wetter or less seasonal climates, you have a much larger variability in the affiliations that you have, suggesting that even if you're tolerant of, or you're more affiliated to drier conditions, you're still able to survive in the wetter conditions, but not the other way around, because here it is only the wet affiliated species that seem to be doing better here in the drier conditions. And when you break this down further into taking the proportions of the driest 20% of species and the wettest 20% of species, that is 20% which shows the strongest affiliations to dry conditions, and the top 20% that show the strongest affiliation to wet conditions, what we find again is that this proportion of these extremely dry affiliated species are really high in the dry conditions, and they dip towards the wetter conditions, but they still stay there. But by contrast, the wet affiliated species they kind of, after a certain measure of seasonal drought, which is about negative 700, which is a measure of water deficit, they just seem to drop out of the system. And even in the systems that they are there, their probability, the proportion that they occupy in the community is much lower, because this community space is now shared by many more species, right? So together we find that when you are dry affiliated, you're actually dominating the communities where you are most affiliated to, but you can still survive in the other communities at a reasonable proportion. And so together, these results seem to suggest to us that dry associated species are persisting in the less seasonal forests as we expected, but not vice versa, suggesting that there's a physiological tolerance that might be eliminating these the wet species from the drier areas. But when we look at the community patterns, what this suggests to us is that if the Western Ghats a place like the Western Ghats is shifting towards more seasonal rainfall, as is predicted to happen, regardless of how much rainfall we get, this is likely to favor dry associated species and perhaps shrink the ranges of wet associated species, which can reorganize the community at these large spatial scales to reshape the biodiversity gradients that we know today, right? And the reason I say this confidently is because we did the same analysis with just precipitation, the total annual precipitation as a predictor. And in all cases, it was the climatic water deficit that seemed to best explain what was happening to these species and these communities in terms of where they were found and how they tended to co-occur in a community. And some of these patterns were also related to species traits. Now, we would, in plant ecology, there are 4 fundamental traits, as I mentioned, one of these previously, seed size, that sort of seem to relate very strongly to plant performance under different conditions. One such trait is called specific leaf area, or here I mention it as SLA. So SLA just means the higher the value of SLA, you have thin, cheap leaves, and low values means you have thicker, denser leaves, which tend to be more stress tolerant, including tolerance of stresses like drought and shade. Now, if you are looking at SLA, it might help you either tolerate drought or it might help you tolerate shaded conditions by having a conservative resource use strategy. If you have thick, dense leaves, you're just investing in staying in there, tolerating stress, and just, being good at hanging on. So when we associate SLA with the species distributions that we modeled, very interestingly, we found that low SLA or thicker, denser leaved species actually very prominently became abundant, as you will see with this darkest line here, in the driest conditions. And this was a very prominent trend, which we didn't quite expect in this, we thought the low SLA species would be more abundant towards the wetter sites where it's very shaded and photosynthesis would be very limited. But instead, it looks like SLA might have something to do with dry tolerance. And this is something that we try to get into a little bit deeper in a more experimental framework using seedlings. Now, I apologize, this is a crowded slide, but I just wanted to bring your attention to two things, which is how traits shape the growth of seedlings, which are these bunch of graphs on the left side, and whether this is related to the photosynthetic rates or the change in photosynthesis that seedlings undergo when they are placed under a drought stress. So if you would just look at the graphs on your left, we see that as expected from the previous results that we see, when you have these high SLA species are actually, I'm sorry, can you see my cursor? These high SLA species seem to be undergoing a more substantial decrease in their growth rates, in their performance, when they are in dry conditions, which is marked by the red line here. Compared to the low SLA or these thicker leaf species, they have low baseline rates of growth, but they just sort of hang in there. There's not much difference that happens. And this might be one reason during the seedling phase, at least, why we are seeing this affiliation of these thicker, denser leaf species towards these drier forests. But despite that, what we found was that SLA was, in an experiment, SLA was actually a poorer predictor of plant performance compared to some of the other traits like the size, anatomical traits like the xylem diameter or the stomatal density, which are more directly related to a plant's physiology of water use, right? And this is what was seen in the physiological responses that we measured to the right here, where we found that it was these anatomical traits which did really well to explain differences among species in their photosynthetic rate when they were subject to drought, as shown in the red line, compared to when they were in well-watered conditions represented in the blue line. And so we would like to now take some of these findings forward to understand. This was just a bunch, set of species in a greenhouse, but we want to take these findings forward to understand what happens to these kind of species, because how do these traits related to anatomy or traits actually related to water use, the physiology of water use and physiology of light use, how do they govern which species do well in the different conditions of light and water availability that we see in forest fragments? So now I've come back to fragments, right? So my reasoning here is that when we take a patch of forest, any patch of forest, conditions tend to be warmer and drier at the forest edge. As you break this patch into smaller parcels, what you're doing is increasing the proportion of these warmer and drier conditions compared to the cool, humid interiors. So you're immediately changing the balance of which species is likely to do well, or how much area might it have to do well in this fragmented forest context. And we think that it is going to be the traits that will drive these responses of species. So we have set up this network of seedling plots. This is all work in progress, but hopefully I can have some results for you at another time. But we have set up this network of seedling plots arrayed from the edge to the interior across this human modified landscape to assess or to link how species traits related to their anatomy, to their physiology, and to their general morphology might explain the performances of these different species as they go from the forest edge to the forest interior. And in the long term, do we see certain trends with what kind of species are doing better at these warmer edges versus the cool, humid interiors? But also, if you take that into an even longer term perspective, if we have climate change happening and these seasonal drought conditions intensifying, the impact of those conditions is likely to be stronger at these forest edges that are already warmer and drier. So the next goal is to kind of see whether we are likely to see some interactions between a changing climate and these spatial gradients in environmental conditions that we see created in forest fragments. So we've started this work. We're going to measure a whole bunch of traits. I'm really excited about it. As we speak, my students are getting ready to measure traits on these seedlings that we are growing. And hopefully we can have some interesting results in some time soon. But overall, what I hope I have convinced you here, or try to make a case saying that if we understand these different abiotic and biotic processes that govern how species interact with each other and the environment, the basic premise of ecology, this is fundamental towards understanding how biodiversity is likely to behave and what is likely to be the trajectory of biodiversity in many human modified systems. So we have these big questions of, are these ecosystems stable in terms of, will there be a lag in how biodiversity is lost? And assessments using recruitment rates and putting it in the framework of species coexistence can help us answer some of these questions. It can also help us have more like an applied perspective to understand habitat quality. What do we mean by habitat quality when we are talking about disparate species with disparate requirements, but they need to coexist together as a community, and give us some handle, hopefully, and what we need to measure and monitor in these systems for a better understanding of mapping the trajectory of biodiversity change in these human modified forests. So this is a big ask, and I hope I can continue some of this work. But as of now, I hope that this, whatever I'm doing and what I've presented to you has helped us at least understand a little bit about what might be the forces that are shaping biodiversity in the Anthropocene as we see them. And I have a lot of people to thank for the work we do. I have fun. Not everybody in the lab is there in this picture, but this is just to show, at our field site, we have a great team, and so much of this work is being done by students. And we also like to have fun while doing this. And with that, I am ready to take any questions.